Let's see. So generally, I guess those of you who heard me before know what my interest in is to understand how things are coordinated across multiple scales, on a molecular scale from your Neo, and on a organismic scale, which we'll talk about today, and also in communities, which is what I guess the focus of today's talk is this heat shock response, which is a fantastic stress response pathway. Basically, what it's supposed to be is to deal with stress on the proteome of the cell. And so that's a major challenge because the proteome, generally proteins in the cell are just a few kT away from a lot of them from electron changes. We good now? Yeah. Oh, beeping. It's making noise. All right. Well, I'm yelling. All right. So the proteome, right? So a lot of environmental signals, and in particular temperature, are affecting the proteome and can destabilize proteins in the cell, make them misfold or aggregate, and this can be highly toxic to the cell. And so because high temperature is one of these inducers, this is collectively called the heat shock response, but there are actually other ways to induce it. And it is, of course, very important stress response pathway, and so it's highly conserved from bacteria to plants to human. And I would call it functionally conserved across kingdoms, and even at the level of molecules, the players are conserved from yeast to human. And so let me very quickly explain how this thing is believed to work. So there are really two types of players here. There are these things that I meant here are denoted by P. These are heat shock proteins. Usually these are molecular chaperones that go and bind protein or misfolded protein and either stabilize them or tag them for degradation. So even under normal conditions, there is some level of heat shock protein that is supposed to maintain homeostasis of the proteome. And then there is this heat shock factor that I denoted by F here, which regulates this pathway. And so the way it works is that heat shock factor HSF is inactive in a monomeric form, and under normal condition, this monomeric form will be bound to one of these heat shock proteins, which will prevent it from tetramering and from being active. So under normal condition, all the heat shock factors in the cell are titrated by heat shock proteins and are inactive. But then when stress comes in, a lot of misfolded proteins start accumulating. These heat shock proteins will go and try to fix the problem. And when they go away in this direction, they allow the heat shock factor to tetramerize, get activated by phosphorylation and methylation, get into the nucleus, and induce the transcription of a large array of heat shock proteins that are now can now go and fix the problem that keeps accumulating in the cell. Okay, so there is this clear positive feedback, and actually there are multiple feedback loops here. And because this system is very important and highly conserved and also very interesting because it has all these feedbacks, it had been modeled multiple times. You can find quite a few modeling papers in the literature looking at the heat shock response in single cells, in bacteria, and in yeast, and in cell cultures, and what have you. And people used all sorts of languages to model it, from dynamical systems to statistical mechanics, and even control theory and stuff like that. And what's common to all these models is that they are very, very detailed. All of them have many equations. There's a lot of parameters, kind of like, driven by pressure from biologists to be as accurate and faithful I guess we heard some of this pressure here yesterday about knowing all the details of the system before you model, etc. And so the question that we ask is moving away from the single cell and into the multicellular context. We ask how this heat shock response is coordinated in a multicellular environment, and specifically here in an animal. And so, on one hand, people until very recently believed that because this whole system is conserved from single-cell organisms to animals, it is probably, and because the stress on the proteome is a single-cell problem, people believe that this is a cell autonomous system, so each cell decides how to maintain its own proteome. But in a multicellular organism, it has a lot of organismic level functions, including behavior. It stands to reason that somehow this system is coordinated, and that's basically our question. So the question we try to address is, these three things that you need to do in order to respond to temperature. So actually to sense temperature, to decide if temperature is high or low, if there is a problem or not, and then to respond. We ask this question: is this, really a cell autonomous problem, or maybe it's coordinated between cells? And in an animal, it can also be a centralized decision, somehow decided by the brain and executed by the cell. Okay, so how do we approach this question? We try to follow the dynamics. So we do this by focusing on C. elegans, the worm. I'm not going to tell you too much about the advantage of the worm, or try to sell it to you once again. But of course, for a physicist, a one-dimensional spherical one-dimensional worm is a nice system to work with. And important for us, it is transparent, and so we can look and see what's happening inside. The problem with working with worm, as I tell you every year, is that worms tend to crawl away when you want to look at them. And so our approach is to trap them in these microfluidic traps. And you can see the worm is kind of it's not going anywhere. You can look at it for a long, long time. Because time here is in hours and minutes. And you will see that it's soon going to lay an egg. Yeah, perfect timing. So the worms are really happy in our devices, and we can track them and look at what's going on for a long, long hours, if that's what we want. And our device can hold a few dozens of worms simultaneously, so we can even generate some data with some statistics. Okay, so here's what the data looks like. We specifically look at some stress risk reporter, DAF16 or FOXO. And what you need to know in order to understand this movie is that DAF16 is cytoplasmic when it's inactive, and it is nuclear when it is active. And so we start this experiment under normal conditions. You see it everywhere. DAF16 tagged with GFP, so it's completely diffusive, mostly in the cytoplasm. And when we'll activate heat shock response by elevating temperature, it is supposed to become nuclear, go into nuclei in the entire worm, and you'll see there's no problem. This thing here is going to be turn red when we heat shock it, and go back to blue when we need to stop. And so I think that's enough to understand what's going on. Oops. If you can actually do it enough. All right. So here's the worm, it's being heat shocked. As advertised, DAF16 run to nuclei. And now it's called again, but it takes some time for DAF16 to go back to its relaxed state. So it takes some time. Time here again is an hour and a minute. Hours and minutes. All right. So first, of course, we can do this experiment at multiple temperatures. And so first I need to tell you that worms live comfortably between 15 and 25 degrees. We cultivate them in this experiment. We cultivate them at 24 before we heat shock them at these different temperatures. And the image that you see here is kind of at the peak of the response, the point where we see the strongest activity, and we subtract the background, so the diffusive part of DAF16 disappears. And so you see, at 80 there is very little response. You can barely see a little more response at 30, and the temperature is definitely increasing as we go up in temperature. And you see that it happens pretty uniformly, right, across the worm. But I can actually tell you, if you look closely, that different tissues of the worm actually respond at different temperatures. So for example, by 31 degrees, the entire intestine of the worm is completely activated, but body wall muscles, which are these small cells over here, are not activated. And then here, the hypodermis and the intestine are activated, but the muscles are not. And here, the entire worm is already light up. So different tissues have different temperature thresholds for activation of heat shock response. Another thing that one can see, you could in principle see so you had seen it in the movie, but let's just notice it now, that the dynamics of activation starts in the head and goes to the tail. So this is when we activate. This is faster than you saw before. This is time in minutes. And you see that the first cells to respond are cells in the head, and then later it propagates here. These cells are all activated, but the rest of the worm is not. And then it propagates backwards. Here it's activated down to here. Finally, after 25 minutes, the entire worm is activated. And this goes in the opposite direction. Now we are cooling the worm off, but you see that the signal again propagates from head to tail. So the first cells that turn things off are in the head, and it goes backwards. The last cells that are still active after 5 hours are the cells in the tail. Okay, so the dynamics always goes head to tail, both on the way up and on the way down. And actually, we see 3 types of dynamics. So let me just explain these figures. They're pretty easy. So what you see, like this axis is the worm head to tail, head on the left, tail on the right. And this is time in hours. And what we do is we take the worm, we divide it into 40 segments, and in each segment we just count how many cells got activated, and this is color-coded like this. Okay, so black means no cells activated in this segment of the worm. And very bright means 6 cells are activated in the worm. 6 and more. Some of these segments have maybe 8 or 10. All right. So what you see here at 31 degrees, you see this dynamic that I was talking about from head to tail, right? So you see that these cells get activated earlier, and then it goes backwards, and these cells activate. This part of the worm is get activated late, and you also see it here in the way down, right? These cells get deactivated first, and these cells get deactivated later. At 32 degrees, you see somewhat different dynamics. On the way up, it still has this head-to-tail dynamics, but you see that now it's on the way down, it's way more persistent, and activation is pretty sporadic. And at 33 degrees, this dynamic disappeared completely. The entire worm just lights up immediately, and again, like very, very persistent response. Look at some cells are active even after 11 hours. And these are specifically these intestinal cells. All right, so kind of phonologically, 3 types of dynamics. The one that is there's a progression, and this is true for all temperatures under 32 degrees, below 32 degrees. And then at 32 degrees, we still see these dynamics, but persistent dynamics on the way down. And at 33 degrees, the worm just lights up immediately. All right, so if you remember, my question was about coordination and centralized response. So here is our attempt at asking, is the response centralized? And so, this is about who senses temperature and to what extent neurons are playing a role here. And so there's a bunch of neurons in the worm that are heavily reported to be somewhat related with temperature. This is mostly in the connection with a behavior called thermotaxis, where worms just go towards a favorable temperature. About it later, or Matt can tell you about it. A paper from, 7 years already suggests that these neurons are also involved in heat shock response. And so the worm is a fantastic animal because you can go and kill these neurons in multiple ways. And so here's our attempt. This is wild type again, it's very high temperature. But if we take any one of the 6 sensory neurons that have been reported to be responsive to temperature, none of them change the heat shock dynamics. So none individually, one by one, if we kill these heat shock temperature responding sensory neurons, none of them has any effect on the dynamics. But this neuron AIY is an interneuron which gets signals from all of these sensory neurons. And so when we ablate this neuron, we see a completely different response, right? We lose this immediate activation, and the persistent is much more sporadic. Some parts of the worm are not even activated. So this is very high temperature. When we kill this interneuron, we see a strong effect on the dynamics. And if we do it at 31 degrees, there will be no response at all. You can always ask what in the neurons can sense temperature, and so the worm expresses one species of temperature sensory receptors, GPCRs, that are major temperature sensors in mammals. And so this is the worms version of the TRPV channel. And when you kill OCR2, the gene that expresses these channels, you basically kill the response. So neurons definitely have a role in this story, right? If you completely kill the ability of neurons in the entire worm to sense temperature, then you kill this heat shock response. Okay, so we also ask ourselves, neurons have some role to play, but what about coordination between cells? Or does a cell have anything to say about is it going to activate heat shock response or not? And so, so an experiment that we can do, because we are in this macrofluidic setup, is to not heat up the entire worm, but only say heat up half the worm. One thing that is true about our device, and we originally thought is a problem, but turned out to actually be a utility, is that we have no control over the direction at which worms get into the device. So some worms will go head first, and some will go tail first. And this turned out to be completely not important if the entire device is uniform, homogeneous. But in this experiment, where we only heat up half of the channel, then some of the worms have head first, and the head gets heat shock, and some have tail first, and the tail has heat shock. So what I'm going to show you now is data where we collect first only the ones that give their head in the worm region. And so this is the head of the worm. This is the region which we raised to 33 degrees. And you see that these cells in the posterior basically do not get activated. So really only the cell, so again, you see these dynamics head to tail, and it's really only the anterior part, the part that sits, the head part that sits in the worm temperature that gets activated. And then there is very late, there is some residual activity in the tail. And then what happens if we look only at the worms that get their tail heated up? So 60-something percent, between 60 and 70% of our worms actually don't respond at all. So if we hit them only in the tail, we don't show any response. But some 40% show response like this. So they do show some response. It is definitely much lower than you'd expect for such high temperature. But you will notice that despite the fact that we hit them in the tail, the dynamics, again, starts in the head and goes backwards. Right? But you will notice that in the head, it is really just neurons that get activated. And very few cells in the region that is not being heat shocked. All right, so dynamics still goes from head to tail, and really you see activation mostly in cells that are really hot, plus head neurons in the case of heat shock in the tail. Let me just skip this. This is really biology. So this is more or less the data that we have. And I remind you that our question is about coordination and centralized control. And so can we understand these data that I showed you in the context of how cells are coordinated? And so I already showed you this cartoon of how the heat shock response gets activated, right? So the idea is that heat shock factor is titrated by heat shock proteins, but these heat shock proteins have nothing better to do. But once there are these solid proteins, heat shock proteins will prefer to go in that direction and release the heat shock factor to go into the nucleus in the active form and make even more heat shock protein. So, in such a small model, you can write way less than 30-something equation and use way less than 20-something parameters. But most importantly, this is a model that is simple enough to actually analyze. And one can easily identify that it has properties of an excitable system. So an excitable system, those of you who don't remember, are systems which have one fixed point that we can call homeostasis. And when you kind of like perturb the system away from this homeostatic fixed point, they will move around the fixed point and will get back to its fixed point. But if you make a longer or persistent perturbation, then still there is only this fixed point, but together will not be a local event, but actually a trajectory that kind of like spans space. So these kind of systems in our field are usually used to model neurons, right? And this trajectory in phase space will be related with the spike of the neuron. So this model bears a lot of similarities with this kind of excitable states. And let me just explain, like hand wave my way through why this is an excitable system. So this is just a cartoon, right? This is no data. But the idea is that during homeostasis, you have very few Hitchcock proteins and very few active Hitchcock factors, and so everything is fine. But now there is induction, and during induction, Hitchcock proteins make this Hitchcock factor free, so there's a lot of active Hitchcock factor, which generates a lot of Hitchcock proteins. Now these Hitchcock proteins, now at huge concentration in the cell, go to deactivate the Hitchcock factor, so there is still a large pool of Hitchcock protein, but Hitchcock factor is turned off, and so no more synthesis of new Hitchcock proteins. And so now Hitchcock proteins will now be slowly titrated by misfolded proteins or turnover. So finally, this will take some time, but finally they will go back down to the homeostatic level. But if the heat shock is still there, then we'll just go back here and have another cycle, right? Another extinction cycle. So there's of course a prediction in this model, and this prediction is that the time scale for the Hitchcock proteins must be much, much longer than the time scale for Hitchcock factors. And again, the worm is transparent, so one can directly look at this Hitchcock factor in the activated form. Maybe not easy to see from where you're sitting, but activated Hitchcock factors form these granules in the nucleus. And so notice that here the Hitchcock factor is very diffusive, and here it's very granular. And so if you just measure this Hitchcock factor star, the active form of Hitchcock factor, by the granularity of the nuclei, you can have some estimate for how much of the Hitchcock factor is active. And so you can see this is like the dynamics of the Hitchcock protein, very slow, and this is the dynamics of the Hitchcock factor at two different temperatures, kind of like the data version of this cartoon. And so maybe that's my message about modeling, right? Is when you have a simple model, you can actually understand what's happening. When you have 30-something equation, it's maybe a little harder. So, okay, so these are about single cells, but again, we are interested in interacting cells, and so how do cells in the worm interact? There are really two ways. One is through this neuronal signaling, which is basically what I showed you. And I showed you that the signal goes from the head and propagates to the tail. And there are also local interactions, and these local interactions come from Hitchcock proteins being able to signal to neighboring cells. And this is very local interaction, right? So Hitchcock proteins lead to secretion of some vesicles that are then sensed by neighboring cells. And so there is this global mean field interaction that propagates from the head to the tail, and this local interaction that are kind of homogeneous in the worm. And if you want to put this into the model, then the neuronal signaling affects this activation, the rate at which the Hitchcock factor gets activated, and this is through this methylation and phosphorylation cycle that I alluded to earlier. And of course, these local interactions come from activation. It comes from measuring the level of the Hitchcock protein and goes to activate this loop in a neighboring cell. And so you add these two ingredients to the model, and you just do it we did it in a linear, non-dimensional version. Excellent. Just placing the cells one after the other in an array from head to tail in the worm. And we have this neuronal signaling go from the head to the tail, and the local signaling between neighboring cells. And this is more or less what these kind of simulations give. So at low temperatures, neuronal signaling leads to activation from head to tail. This is what we see at temperature that are 31 and below. At high temperature, the neuron initiates the activation, but the local interactions dominate the dynamics. And so they do two things. First, they make this activation much faster. All you need the neurons to do is to initiate this cascade, and the rest happens due to local signaling. And the other thing that they do is to stabilize this response, as we've seen in the data, because if you think about it, now this local signaling is kind of like acts as an auto-inducer, right? So you induce your neighboring cells, and they induce you, and locally you generate in such it's not really stable state, but it's definitely persistent. If you don't have a neuron, then you don't have this progression from head to tail, and the activation is sporadic. It's kind of what we saw in the OCR2 mutant, the one that does not have neurons playing in the story. And finally, with alcohol induction, these cells that are in the cold region will not be activated nothing to make them excitable. So just in the language of excitable system, this system is an excitable media, right? The signal can, once one cell is excited, it can excite all the excitable cells downstream, and this accounts for the entire dynamic. Okay, just said all these. I just want to say passing one thing I didn't mention is this idea of temperature threshold, which is that there is one kind of response up to some temperature, and then a very sustainable response at high temperatures. That's something that is very well known in mammals, and specifically we humans are very capable of sensing temperatures to a fraction of degrees up to 50 degrees. And after 50 degrees, we only feel hot and painful. Right? So this is the idea that there is a threshold, and that beyond the threshold, some new dynamic thing is actually everywhere, and here we at least have some explanation. Okay, with this I want to thank people in my lab, collaborators, and to advertise shamelessly that we are looking for courses, so either the theory or experiment involved. And thank you all for sticking around so late.
When you do your modeling, what's the time scales for the communication between the neurons compared for the cell communication?
Yeah, right. So that's for the most important thing, right? That these time scale these. So what we learned from the experiment is that from head to tail, the signal propagates in 30 minutes. And that sets the scale for this neuronal signaling. And the slide that is here is a slide where we show that a particular hormonal molecule in the worm that is known to work roughly the scale is probably responsible for this propagation from head to tail. It's a giant kinase pathway. The timescale for these local interactions we assume to be almost instantaneous. So since there is no, in the signaling itself, there is no protein production or anything like that, it's just an export of molecules that are already in the cell, then I would say it probably should be on the order of a few minutes, and that's roughly.
So the other thing you said is that the local excitation is much stronger than the other one. The other one just sort of give a small kick and then the system moves. So when you hit the tail, you expect much lighter and much lower excitation on the head. They appear to be similar. Or that
okay, so so there is something misleading in the way they represent the data here. What happens when we hit the tail, the only thing that gets activated in the head are neurons. Only neurons.
That's correct. That's correct.
Somatic cells are not activated there because locally they are not excited.
That's why I was wondering why they expect much lighter excitation on the head than on the tail.
That's right. So that's
the case of figure.
Yeah, so that's exactly. That's because there is in the head there is this nerve ring, which is a very dense collection of neurons, and sort of way more spread in the tail, and so it just makes this figure misleading.
And just to finish up, and when you put the numbers also get like that half the time on the side of the head, and half the place.
Yeah. So okay. So there are two questions for us about this, which is, clearly the head needs to know somehow that there is a problem in the tail, otherwise neurons will not kick in and nothing will get activated. So how is this information propagating from the tail to the head? We actually don't know. One hypothesis is that some of these sensory neurons actually have processes that go all the way to the tail, so maybe this is the way this is sent, really directly. Another possibility is that there is some signaling molecule going back from the tail to the head, but we don't know. It's really interesting that in the worm, all the neurons that have ever been reported to be activated by high temperatures are located in the nose, and the only temperature-sensitive neuron that is known in the tail responds only to cold temperatures.
Any other questions? If not, let's go to